Hello, welcome dear students. This is Dr HADI here and you are watching Medical Globe. Today's topic is from biochemistry, that is tertiary structure and quaternary structure of protein. We have already discussed the primary structure in detail in one single video lecture, and then secondary structure was also discussed in second video lecture. In today's lecture, we will discuss tertiary and quaternary structure along with super secondary structure as well. So it will be a short video lecture. I hope you will enjoy it. So let's start. In order to understand this concept of tertiary structure, I would like to draw a diagram here. And let's suppose this is the diagram. That is a diagram. So I'm going to write it like this, is moving like this. Okay, we said that when a polypeptide, this is a polypeptide, it is straight chain polypeptide, and it makes some sort of bend, twist, it is called a secondary structure. So this is already discussed, secondary structure. Now see, here once again I'm going to write, this is actually secondary structure. This is a secondary structure. But if you see here, that secondary structure, it is already folded, it is already twisted. Now it is going to refold upon itself. Now this is become like this. Now it is going to refold upon itself. Let's see, now it has become refold upon itself. So when a polypeptide become like this, it fold and refold upon itself, then a three-dimensional structure is formed. Now this three-dimensional structure will be something like a globular thing that you can see from front, side, behind, back, top and bottom. So a three-dimensional structure will be obtained because of this refolding, and that structure will be called as tertiary structure. Look, when it was not refolded, I mean, if you see it only here up to this place, if you see here, that is only secondary structure. That is only secondary structure. But when that structure fold and refold upon itself, then we call it tertiary structure. Okay, fine. In the tertiary structure, you will see that there is a surface, and this is the core center. Let's suppose if I write, if we mention the tertiary structure of protein like this, so that region will be called as the core, the central part of that protein, and that will be the peripheral part of the protein, globular protein. These here you will find, and these region you will find hydrophilic amino acids, and here at the core, the center, you will find hydrophobic amino acids. And we know very well what are hydrophobic amino acid and what are hydrophilic amino acid. We know very well about these. Okay, then the question comes, what are the bonds that are present in the tertiary structure? Just like we discussed the bond in the secondary structure, that was beside the peptide bond, there was hydrogen bonding. In the tertiary structure, the bonds are besides hydrogen bonding also present. So hydrogen bond, this is the already present as it was discussed in secondary structure. Fine. What are the new bonds that were not seen in the secondary structure? That is okay, disulfide bond is also present here, disulfide bond, and ionic interactions that was not mentioned in secondary structure. And then another, we have hydrophobic interaction, this is, and van der Waals forces. So these are the name of the bonds that are present in the tertiary structure, and that they give stability to that structure. Now question comes, what is mean by disulfide bond? A disulfide bond will always be in between the sulfhydryl group of one amino acid and the sulfhydryl group of the another amino acid, SH. When they make bond, then become S, then it become S, and H will be removed. So this SS bond is called as disulfide bond. Ionic interaction is in between a positively charged species and a negatively charged species. Maybe if you see here that a positive charge develop on some of the atom in an amino acid, and negative charge develop on another atom of the amino acid, then a bond will be formed between a positive negative charge. That will be called as ionic interaction or electrostatic interaction as well, because in amino acid we can see positive negative charge, as this is possible because of the removal of the electron or addition of the electron. We can see this is quite possible. Then we have hydrophobic interaction. The scientists usually do not consider the hydrophobic interaction as a bond, but it is a sort of interaction that is created because of the propulsion of water. Means water propel them. What? It is actually between the side chain, especially like methyl group and methyl group of another amino acid. As methyl side chain is not soluble in water, so the water will repel this side chain. It will be repelled from that side, it will be repelled from that side, from that side, from all around, all the sides. The water will repel these methylated groups or side chain of the amino acid. As a result, the side chain will not go away from each other, and they will cling together. They are not coming together by their own will, but something else from outside is pushing them to come close. That is why this kind of bond is called as a hydrophobic interaction. There is no bond that's like we say in CH3 there is a single covalent bond. No, this is not actually the bond. This is actually called as hydrophobic interaction. Hydro means water, because these two groups are bring closer because of the water, and that's what's called as hydrophobic interaction. And this is so important thing. Most of students, they skip this point. They really don't know what is meant by hydrophobic interaction. They know the word hydrophobic, water fearing, but they actually don't know. Then there is another thing that is van der Waals forces. Van der Waals forces are also, as we know, that if there is one molecule and there is another molecule, sometime because of the distribution of the electronic cloud in these molecules, a charge may be produced at a temporary charge, negative or positive charge will be produced, that we call them a dipole, and then as a result of this charge a bond is created, which is called as van der Waals forces. So we also discussed the bonds. Along with the name of the bond, we'll discuss what are these bonds actually. Okay, now what is the example of that tertiary structure is the myoglobin. Myoglobin is an example of that tertiary structure, myoglobin, and this is a protein just like hemoglobin, but it stores and carry oxygen, especially for the muscle, so we will write it muscle. Which muscle? Skeletal muscle. Maybe they are also found in a smooth muscle, but especially they are found in a skeletal muscle where they provide oxygen and store oxygen, give it to the muscle. Then we come to the quaternary structure. Oh my god, before we go to quaternary structure, let me discuss something, one thing more, that is supersecondary structure. So something new, this is not mentioned in lower classes like FSC level, but is mentioned in advanced classes of biochemistry, master level biochemistry. That is supersecondary structure is what I would like to tell you. That what is that supersecondary structure? A supersecondary structure. According to the book of Satyanarayan, supersecondary structure is the intermediate form of protein. Intermediate form of structure. Intermediate between what? So when the word come intermediate, so it will come between two things. That is secondary structure and tertiary structure. Means in between the secondary and tertiary structure there is another organization of protein, that is, we call it supersecondary. Here its position is here, mean it will be a little bit more advanced than the secondary and less advanced than the tertiary structure. What is there inside that supersecondary structure? If you see inside a supersecondary structure, you will see that there will be alpha protein, alpha helix plus beta pleated sheet. Both these type of protein will be found, and these two will be connected by the help of some loop. So when a loop connect alpha helix with the beta pleated sheet, this sort of protein will be given another name as a supersecondary structure. And overall, this kind of combination that we call supersecondary, it is also called as motif. Means sometime if you see that word in biochemistry, what is mean by motif? Motif represents the combination of alpha helix with beta pleated sheet being connected with one another through the loops of protein chain, and we call it motif or supersecondary structure. So that's gone. Now come to the last one, is quaternary structure. Here quaternary structure is also present here, and now it's more easy than the tertiary structure. What is quaternary structure? In quaternary structure we will see an association of two or more than two, this kind of unit, like this tertiary unit, like this is a tertiary unit. Let me write here very clearly on the whiteboard. If I see, let's suppose that this is one tertiary protein, a unit of tertiary protein, and then there is another tertiary protein, another unit, 2, 3, 4 or whatsoever. When these two units are connected with one another, then this will be called as another advanced organization in protein, another advanced structure, that is called as quaternary structure. So there must be some bonds that will connect these tertiary structures with one another. And do you know what is the name that we give to this kind of protein? That we give the name oligomers. Oligomers. Just like we give the name motif to the supersecondary structure, we give the name oligomers to the protein in which tertiary structures are combined. And if two units are combined, we will call it dimer. We will call it dimer. So that word dimer you may see or hear in some of the books, that there is an enzyme, it is dimer. Means the enzyme consists of two parts. 1, 2. There's a trimer, a tetramer. A tetramer means that will contain 4 units. 4 unit. And each unit is definitely a tertiary structure. So many tertiary structure linked together by some bonds. Of course there will be some bond. What are these bonds? Actually hydrogen bond, okay, maybe hydrogen bond, the maybe ionic bond, and hydrophobic interaction. These 3 bonds are found in the quaternary structure, and means in between these units. So if we take the example, what is the example of quaternary structure? Is there any protein? So we say yes. Hemoglobin is a very famous example of that. Hemoglobin. Hemoglobin is a very famous example of the quaternary structure. Why? Because in hemoglobin you will see one three-dimensional tertiary structure, second three-dimensional tertiary structure, third and fourth. These four chains are connected with one another, and we give the name hemoglobin to it. Now, one of these two of these four chains are called as alpha chain means alpha chain, and remaining two are called as beta chain. Alpha chain and beta chains. And we have already discussed the number of amino acid, 574 amino acid, 141, 146 amino acid in each. The structure of that hemoglobin has already been discussed in the primary structure of protein, where we discussed how the primary structure is important in reference to the sickle cell anemia. So here we go. We got all the structure: primary structure, secondary, tertiary, super secondary, and finally quaternary structure. We discussed all. I hope that you enjoyed that video as well. And if some students wants to have a complete clear idea of all the structure of protein, I will suggest that please search the first video on primary structure. But before primary structure, search the summary or overall, where I have discussed overall the primary, secondary, tertiary and quaternary structure in just one video. That was the summary. And then slowly we discussed one lecture of primary structure, then we discussed another secondary structure, and then tertiary and quaternary structure. I split, as I told, I split that my whole topic in 3 lectures or 4 lectures, just for the convenience of the students, because if I make it a video of 1 hour or 1.5 hours, maybe some students get bored and they don't like such lengthy videos, lectures, especially in a condition like prevailing nowadays, corona. Yes, every day, every day we have a corona disease. So we pray to God that immediately, as soon as possible, this disease disappear from the earth, and our grounds, our picnic points, our universities, colleges, all become open once again, get open once again. So thank you for watching the video. Give us a like and also subscribe and share the video. Bye bye. Goodbye.